Good morning, everybody. So we are starting this webinar. If you're just joining, please do switch off your camera and microphone during the presentation. We're going to record it so that people could use the advantage of re-watching it later on if they want. So it's a pleasure to do this webinar. I mean, with the COVID situation, it becomes kind of a useful and essential tools to our activities. We also wanted to do this before the meeting that we'll have in June for the Genome Center. And we want to highlight some really cool activity that is going on here. So I would like to first give you a bit of an idea of the agenda. We're going to have an introduction by the director, Dermitzakis, in a few moments. And then we're going to present to you the status of the DNA sequencing platform led by Harshman, to also give you some really good news that is emerging over the last few months. And we have to have good news these days, as everybody knows. We're going to have after a presentation by Katrin Männik, our head of data strategy, on the data and the governance platform that we are setting up in order to sustain and help the clinical research and basic research here at the Genome Center. And as well, we're going to try to give you a bit of an update of where we are for the analysis and interpretation platform. We conclude and have a Q&A. We have around 160 people that registered, so obviously we will not be able to answer all the questions, but we'll try to select questions. If you have, you can actually write them into the conversation box, and we'll take them at the end and see which one we could answer. And if we cannot, we're going to contact you by email when necessary. Without further ado, let me introduce to you Dermitzakis, who is the director of the Health 2030 Genome Center, for a short introduction. Manolis.
Okay, thank you very much, Ioannis. Thank you very much for joining. I think this is actually an important opportunity to connect with you, and as COVID is starting to kind of be under control, and we're slowly getting our lives back. We're thinking more about exciting projects and things to do using the infrastructure that have been set and put forward by many efforts. As you probably know, but I'm going to reiterate, the Genome Center is a hub to promote genomic medicine. That was the basis of its foundation. And actually, the foundation itself relies on the support of multiple institutions who are supporting this both administratively, financially, as well as intellectually. As you can see from the slide, there are 7 institutions currently supporting the Genome Center. That's the EPFL, the CHUV, the University of Lausanne, Inselspital, University of Bern, University of Geneva, and the HUZ. Now, the whole idea behind the Genome Center is to actually not only provide sort of a general scope and vision, but to actually create the appropriate infrastructure so that all these things can be achieved. And by that infrastructure, we certainly do not mean, and we never intended this to be sort of a place where everything happens, but more of a place where things are triggered. As a place where we can start discussions, we can trigger and activate and sensitize the environment of Switzerland generally, not just for the 7 institutions, and get things moving in all the different directions. Now, some of the key infrastructures that we need when we talk about genomic medicine is obviously the analysis of the genomes, meaning the data generation. That's why we have a DNA sequencing platform that is essentially at the top of a line in terms of available instruments and technology, led by Harshman, and he's going to give you a short presentation of where we are. And as Ioannis said, there's some exciting news which I'm not going to share. I leave it for Keith to benefit from that, and also the huge amount of effort that he led in order for this to be achieved. And I want to congratulate him and thank him very much for that. But he will tell you what this is about. At the same time, and actually we're very fortunate to have Ioannis leading the data analysis and interpretation platform. This is basically the complementary component required for sequence analysis and genome analysis, which is the data analysis part and the interpretation of data going from research to clinical implementation. You hear more from the different people, including Ioannis and all the other people in the platform. And I want to say that, of course, this platform, the data analysis interpretation platform, was essential and key in order for the good news to be achieved that Keith is going to talk about. I know it's a bit weird to be talking about something that you don't know, but I don't want to steal the thunder from Keith. But basically, we're actually in a very fortunate situation to have two platforms that are very complementary, but also the people are actually working very well in perfect alignment and coordination. And of course, this is to the credit of everybody, but in particular to Keith and Ioannis for having essentially giving the example of how these things work together. The third pillar that we are developing slowly is actually to bring the research community closer, so that this infrastructure from the research perspective can be exploited at the same time that we're building a clinical component. And that's going to be developing over the next few months to strengthen the clinical component so that this infrastructure can serve clinical services from all hospitals in Switzerland and beyond. So I'll leave it here. I don't want to spend any more time because time is essential. And I'm happy to answer any questions or intervene anytime that people think might be necessary. So I turn over to Ioannis so that he can turn it over to Keith.
Thank you, Emmanouil. Thanks for giving kind of a teaser of what is next. So Keith, the floor is yours to present the DSP activity.
Okay, well, thanks very much for the introduction, Ioannis, and hello to everyone. So what I'm going to do is I'm going to present a few slides on the activities of the Genome Center DNA sequencing platform. So I'll refer to it as the DSP, but so what I'm going to do is describe a little bit about what happens upstream of the DAIP activities that we'll hear about later this morning. So on the first slide, I describe just real briefly what the mission of the DSP is, which is twofold. So that's to provide DNA sequencing services and support to first the Swiss clinical diagnostic community, and second, the large-scale genetics and genomics research community. So as you probably know, data generation for clinical diagnostics requires that the laboratory generating the data be accredited for those activities. So the next slide, please. So to this end, what we're doing now is in fact coming to the very end, finishing the process of obtaining ISO 15189 accreditation. So we've successfully completed all the evaluation steps, and this week our application should arrive on the desk of the head of the Swiss Accreditation Society for his signature and final approval. So with that accreditation, the DSP will be certified to produce clinical grade sequencing data. So what that means is that all of the data generated by the DSP using the accredited assays, and I'll describe what those assays are in just a minute, but all of the data generated with those assays will be clinical grade, regardless of whether the sample originates in a diagnostic or a research setting. So I think that's actually quite important to understand. So it's worth repeating. Because they are processed in an accredited sequencing environment, even samples from clinical research projects will produce clinical grade data, which, when appropriate, can be used to inform clinical decisions. So what are the sequencing services that we support? That's on the next slide, please. Up in the upper portion of the slide are the services that we currently support. Since the beginning of our operation, we've supported whole genome sequencing, whole exome sequencing, RNA-seq, as well as sequencing of user-prepared libraries. Since January, we've been quite involved in SARS-CoV-2 whole genome sequencing. This is something that Lorenzo will have quite a lot to say later on this morning. So in the bottom portion of the slide, the point I'm trying to make with this other applications based upon interest and demand is that we're actually quite flexible to implementing and supporting additional sequencing assays in response to the demand of our user community. So I think that's a theme that you'll hear a number of times today, in response to the desires, to the requirements of our user community. So for example, we're now implementing or discussing implementation of a low quantity RNA-seq protocol, and we've discussed the possibility of implementing a microbiome analysis service. So you can find additional information about these services on our website. Information like, well, what's the sample requirements, what's the price, and this sort of thing. So you can go to our website to get that information. So not all of these services fall under our ISO accreditation. The ones that do are on the next slide. So again, whole genome sequencing, whole exome sequencing, RNA-seq. Those will be covered by our initial ISO accreditation. But I don't want to make you think that this will be for forever. The offerings that we'll have for accredited services. We're not going to be limited to this. If the demand dictates, our accreditation can be expanded to include other sequencing services. So I'll finish with a couple slides of about our infrastructure, starting with our sequencing infrastructure. We have a NovaSeq 6000, two HiSeq 4000s. We're also always keen to evaluate other sequencing and complementary genomic technologies if we think they might provide advantages to our existing services, or be able to expand our existing services, or if we feel that these technologies can support useful assays that aren't adequately covered by our existing infrastructure. The next slide, I think, is just a description of laboratory infrastructure. So we have we make extensive use of automation for sample quality assessment, processing, and then of course there's a high capacity, highly secure IT infrastructure for data management and analysis. My last two slides. The next one is a couple photos. In fact, why don't we just go to that's our laboratory and some of the instrumentation. The last photo is my colleagues on the DNA sequencing platform staff, plus the Genome Center Administrative Coordinator, who happened to be on site yesterday when we took the photo. So thanks very much. That's all. That's it for me. I'm happy to ask questions, but let's wait until the end of the session and during the Q&A session that's moderated by Ioannis. So thanks very much.
Thanks, Keith. So we are breaking the news in this case with the accreditation. As we all know, in Switzerland, things takes a bit of time, and I will not take any example, but I think everybody could think of a few. What is also important is that whenever you start to generate data and you generate data at scales, it becomes quite critical to have a data governance and a data strategy. And this is where Katrin Männik is playing an essential role by bridging the Genome Center to the Swiss landscape. And I'll hand over to Katrin for the next set of few minutes to describe where we are at from a data and governance platform. Katrin?
Thank you, Ioannis, and good morning to everybody. So what you can see here is the map of genomics landscape in Switzerland, presenting the academic institutions and university hospitals that are involved in genomic research and medicine, as well as some specialized platforms like us or the Swiss Biobanking platform. And now during the next around 10 minutes, I would like to give you a brief overview. How does the Genome Center position itself and its role in the genomic data governance in Switzerland? First, I would like to underline that we are not operating in isolation. Instead, we are working towards our goals in partnership with several key institutions. As Emmanouil already said, we are a multi-institutional hub to promote genomic research and medicine, and also the genomics arm of the Health 2030 initiative partnered by the EPFL and universities and university hospitals of Geneva, Lausanne, and Bern. On top of that, we are closely involved in the activities of two leading initiatives in personalized health at the Swiss national level. The personalized health and related technologies of the ETH domain and the Swiss Personalized Health Network, particularly through the Biomed IT project. We have recently also partnered up with the Swiss Data Science Center to benchmark their Renku platform for management and sharing of genomic data and started initial collaborative discussions also with the Swiss biobanking platform regarding harmonizing our practices with their experience and also bridging the sample and data tracking levels in genomics. So with excellent expertise in-house and good partners around us, what is the Genome Center aims to provide, both to our current customers, but also to the genomics community in more general? Our goal as a service provider has 4 pillars. First, clinical grade generation and processing of sequence data at scale, as Keith already mentioned. Second, setting up a comprehensive portfolio of analytical pipelines for genomic and transcriptomic analysis. Third, providing means also for multi-omic approaches by integrating genomic analysis with other molecular data types, such as, for example, proteomics or metabolomics. And finally, genomic data lifecycle governance, enabling secure storage, use, and repurposing of genomic and transcriptomic data for both research and clinical applications. So where we are with this plan? As presented by Keith, the first stage of accrediting DNA sequencing platform is now accomplished. And as of the end of last year, approximately, we have gradually started moving towards accreditation of data analysis and interpretation pipelines. So Ioannis will soon talk about that in more detail. And in parallel with that, we have focused on setting up a secure analytical environment for genomic research and clinical decision making, as well as a privacy preserving data governance platform that I'm now briefly introducing to you. So why we think that providing a secure data governance environment is really necessary? So here are listed a few problems in genomic data management that many research institutions and hospitals experience already today. First, of course, the volumes of sequencing data are large. An average sequence of whole human genome is around 200 gigabytes. And if you're thinking now that a regular analysis of Mendelian disease includes hundreds of samples, and the sample sizes nowadays are in hundreds of thousands in case of common complex disease, it's very clear that accumulating amounts of sequence data that needs to be stored securely very quickly becomes a heavy burden. Also, genomic data is often produced to ask only one specific question. And after that, often ends up deposited in out-of-date and unusable data silos. Instead of keeping this once generated sequence data sort of alive through systematic reanalysis in light of either new questions of interest or new incoming knowledge. And side effects of these data silos are often also simply poor understanding of available data and also limited usability because the data itself or its quality are poorly characterized or simply the formats don't match across different data sets. And at the same time, we all know that there is increasing expectation to make data available for reproducible research and for maximizing the benefits that one can acquire from this produced data. And that definitely needs a functional solutions to both facilitate the data sharing procedure for data owners and also to allow access and analysis of sensitive data in a privacy preserving manner. So to meet these needs, The Genome Center has started preparing a platform to offer data lifecycle and storage service at 3 different levels of customer needs, starting from the simplest that we are ready to provide already now to more complex and long-term future directions. But before I will continue, I would like to make one thing very clear. The Genome Center is only an enabler for genomic medicine. In all cases, the customer is and remains the data owner and the controller. So the Genome Center will not analyze, publish, share, or otherwise use any data that is generated or hosted in our environment, unless a specific task is directly mandated to us by the data owner. So the first service level that the Genome Center is ready to offer now includes one-time data processing and analysis by the DAIP. The data are time-stamped and remain as it is at the end of the project. While in case of our regular sequencing service at the moment, our policy is that the Genome Center stores a backup copy of the data for 6 months. Now, in case of this service now, the customer has also a possibility to store and access their data in a secure environment beyond the 6 months limit. So for a longer period that is agreed between the customer and the Genome Center. So this case is pretty simple. We know that there is a high need to effectively store sequence data, and we are ready to meet this need. In case of more complex service levels, we have a general plan for 2 models, but instead of setting up a system and later trying to fit the real life need into it, we are currently exploring what are the most likely data analysis and governance scenarios our customers would indeed regularly request, so that we could, since the beginning, optimize our service according to the real life needs. So in this plan, the second level is a data lifecycle service that we expect to start providing by Q3 of 2021. And this includes the same data storage conditions as the first level, but is meant for customers who want that their project remains active for continuous analysis, with also the possibility to use the Genome Center analysis portfolio and other resources. And finally, our long-term goal is to set up an integrated data governance platform that is not limited to private data sets of particular customers, but allows full genomic data lifecycle management with data sharing, accessing, and linking across multiple data sets, of course, in a privacy-preserving manner. So that here we can truly start talking already about genomic data repository function for repurposing and enhancing existing and also newly generated data. Of course, this is not a service we can come up with tomorrow, but it's a more forward-looking direction that involves also other strategic partners, such as the SPHN Data Coordination Center or the Swiss Data Science Center, and naturally also broader investigation in the community, what really is needed by clinicians and researchers. So this is exactly where we are at the moment. We have started benchmarking for certain technical solutions, building up a provisional concept, and going out to discuss with the community. And with that, I will give now the stage over to Ioannis again, who will explain in more detail the data support, what we can offer today and in the near future. Thank you.
Thank you, Katrin. I think with your presentation, we also show that we are taking the data very seriously. We are setting up the ground for making sure that the data is correctly used under all the best guidelines and proper use, but also that we are actually not trying to develop something that nobody's going to use it. So we're going to really be listening in the coming weeks and months to what the real needs are, rather than developing something that is deployed but not really used or not not really adequate for the real-time needs. Now, let's go into the data analysis and interpretation platform, and I'd like to show a few vignettes for this, but let me guide you through first the people that are behind here. So as you can see, we are all smiling behind our mask. We are a rather small team, but still a very good set of expertise, which I hope will demonstrate during this presentation that we have the right skill set to enable the data analysis and interpretation platform at scale for most, if not all, projects that are required by our panel of institutions that require expertise. Now, one thing that is clear, and we have to set the stage very clearly here, is that we are accrediting, and we are waiting for the document, the accreditation for the sequencing. There are medical use and diagnostic that will come as the next step. And those ones we need to prepare ourselves to design, implement this medical diagnostic for the institution. We will not develop them out of our own interest because we are there to support the clinical research. But if there is enough groups that are interested to implement that, the aim is really to seek CE-IVD for in vitro diagnostic marking for some of these applications. And we might actually select a few, or we might have the problem of having too many and having to choose. And in this case, we are hoping that the governance of the Genome Center will help us prioritize those ones. The second example I'd like, and this will be presented by Ilya on the technology choice that we've made to try to enable this kind of medical diagnostic orientation. The second aspect is something related to the use of the expertise to repurpose ourselves in a very rapid manner to support the SARS-CoV-2 surveillance. And as you've been seeing, the virus is mutating, and having capacity and surveillance capacity with genomic, not just the PCR, but also sequencing, is becoming quite an important aspect. And Lorenzo will talk to you about this in the second vignette. Finally, I'm going to conclude with some example of what we would call high throughput analysis and integrative multiomics, which is trying to make the liaison and the connection between all the omics technologies. We are not unlike the US, the UK, or China, a very populous, we don't have a large population, so we can't really do the 1 million genomes or the 10 million genomes, but we are more into probably the more detailed and multi-omics approach. And I'd show you some present examples of such kind of things. Now, let me hand over to Ilya for him to present what we are trying to do in order to enable this medical diagnostic and the tools that are behind, both in software and hardware, to accelerate this so that we could provide a service at scale for the people that would require our expertise. Ilya?
Thank you, Ioannis. So as it has been discussed today, one of the goals for the Genome Center in the near future is to solidify our data analysis and interpretation offerings to better serve our clients and the community as a whole. And to better see how the hardware accelerated solutions can be instrumental to this goal, let us first have a look at our production infrastructure, its role, and the constraints we encounter with it. So the first thing to say is that the infrastructure that we have is absolutely critical for the internal processes, for instance, quality control, and it is accredited for the production of raw data, meaning FASTQ files. So actually it provides a very high level of automation and scalability, and the standard workflows can be run with minimal intervention, effectively parallelized on the internal cluster. So if you are interested, there is the code is publicly available at the GitHub link below. But the automation, the scalability come at the cost of system being quite complex. And as a result, changing the system or introducing new alternative pipelines into it becomes a rather complex task with a significant, cognitive overhead. And talking about the system, if we think about WGS and WS processing, it is actually based on JTK best practices, which is de facto industry standard. So this is clearly an advantage, but unfortunately, it also comes with the disadvantage of whole genome processing at scale, taking a lot of time, even when we run it in our cluster in a highly parallel manner. So let us see how the hardware accelerated solutions can help us. So first is that such solutions can significantly streamline our data processing in terms of speed. And speed, I think, is becoming very important in genomics, and you will see this for the COVID sequencing. And also, I think at least one of our customers was interested or is interested in rapid turnaround sequencing. And also in the community in the whole, we've seen some very, very advanced project in that space, for instance, in the Rady Hospital in the US. But other aspect is that not only it reduces the time to delivery potentially, but also significantly reduces the support burden, because the operations on to process the data become the scale of it grows. For instance, if you had to run, let's say, 20 pcard and GTK commands to process the genome to call the clinical to call the small variants, so now it becomes much simpler to accomplish this task because the level of granularity is much higher. And another aspect is that such pipelines can internally provide a lot of functionality that we can quickly integrate and offer to our clients. So the examples could be calling of copy number variants, structural variants, or matched tumor normal analysis. And other aspect of this is we've seen some integration between the analysis pipelines and interpretation tools, basically, when you run the analysis and at the same time you are getting interpretation. For instance, variants annotations could be used for research or clinical purposes potentially. So what we've evaluated are two technologies that emerged recently, let's say over the last 5 years. So one is GPU based. It was developed by a company called Parabricks and is currently owned by NVIDIA. And another one is based on FPGA. And it was developed by a company recently, or 2 years ago, bought by Illumina. So now it's Illumina. So the scenario I will talk about today is small variance calling on WGS data. So the graph on the right presents the time of computation versus the FASTQ size in zipped form, which is on the x-axis. So the blue dots correspond to dragging, the red dots correspond to Parabricks running on GPU, and the other two clouds correspond to our production pipeline run on two different computers. So one is faster, another one is slower. And if we look at the typical scenario of processing roughly 30x WGS sample, then on our production infrastructure, it takes about 3.5 hours. If you run it on a slightly faster computer, it becomes 3 hours. With Parabricks in the configuration that we had, it becomes 2 hours. But what is very impressive that when we run it on dragging, it only becomes 20 minutes, which is 15 times faster than our current infrastructure. And it is important to mention that dragging also scales very well as the data grows, and you can see on the plot. So for instance, running small variant calling on 90x coverage data takes only around 40 minutes. And of course, the speed itself is not very, the speed itself, only matters if the results are accurate. So if we look at the accuracy of the small variants that we've seen in the dragging and JTK presented here that were computed on well-known genome on the bottle samples, then we've seen that dragging actually has a slightly higher recall for SNVs, and we see higher precision and recall for indels. So of course, is very marginal because the precision and accuracy even in JTK pipeline are very, very high. So all of this is about 99%. And another point I would like to provide you with an example of annotations or interpretation capabilities of dragging that actually now are being integrated in this hardware accelerated solution. Next slide, please. Oh, sorry. So dragging actually offers quite some functionality on top of the small variance calling. So some of them I already mentioned. This is copy number variant calling, structural variant calling, and all this can be run in trio-based or tumor normal matched pairs. Also, there is functionality for repeat expansion, single cell processing, and methylation pipelines. And finally, recently they have integrated lossless compression, which is very important to us because we are storing lots of data. So if it takes less space, it's very good. And finally, in the integration with the clinical annotation tools, which are currently evaluating on a set of families selected for CNV variants known to be pathogenic for intellectual disabilities in collaboration with the UNIGE. So in the next slide, I will show you how the data looks like. So this is just one example of CNV variant that has been called. And below you see the annotations from Klingen database, and you see that there are phenotypes both in the verbal description and also in the identifiers that can be reused and meshed with other databases. And there's lots of annotations produced like this, and I think with some additional work from our side on integrating and sifting through this data for a particular purpose, it would become very useful for you. So we have decided to go forward with integrating drugging in our processes, and hopefully it will be made available as soon as possible. Yeah, I think that's all for me today. Thank you very much for your attention. And I would like to thank all those people listed on this slide.
Thank you, Ilya. I think what we've seen is that we have also to think about several approaches that are important here. The first one is to get the best technology, and the not invented here syndrome should not apply. And that's why we think that industry grade solutions are great aspect that we need to embark into. The second is that we absolutely require to produce the data in a time to delivery that is essential to our mission. Now, one thing that is also important is that the day you embark into a medical diagnostic exercise, you need to have control over your workflow, and you control also on the way you do the analysis. And this is something that we are taking really seriously, and that will be the future of the DIAP in the coming months. Now, let me go to the second vignette, which is the one from the SARS-CoV-2 that Lorenzo will talk to you about. This is a project of a Swiss surveillance that is led by Laurent Kaiser group at the HUG, that is the Swiss reference laboratory for Switzerland, but also for Europe in emerging zoonosis, of which the SARS-CoV-2 is a good example. And to this point, I let the floor to Luli.
Thank you, Ioannis. I take off my mask as I share the office with Ioannis at this time. Sorry. Yep. I start by presenting the timeline of what happened the last 5 months, basically 6 months. Early December, we've been asked to provide a solution to sequence the full genome of SARS-CoV-2 from a large number of patients. And this is on a weekly basis as for this program of surveillance. So during this month, we set up the sequencing analysis procedures using a panel of samples from HUG and the Tanja Stadler lab in Basel. Then in early 2021, we delivered the first clinical samples, and during the same months, we finalized or optimized our sequencing analysis pipeline. And since February, we processed 752 samples per week, and we have the capacity to double this if required. Just for your information, we sequenced more than 10,000 clinical samples since the beginning of the year. So as I told you, in December last year, we evaluated different methods and technologies. And here is the comparison we made with the Illumina and versus Sophia Genetics Paragon solutions to sequence SARS-CoV-2. Basically, the outcome was that both technologies can be used. There are some differences. Illumina produces a higher sequencing depth, as is shown by the red line on the plot. And Sophia was producing less reads, but it was a bit more robust when samples have an ICT. But basically both technologies were okay. But we decided to move with Illumina, forward with Illumina, as for us it was easier to scale up. First was the time cost. The procedure in the lab were much easier, and so less time consuming. And also the sequencing is shorter for Illumina, which means that we can sequence faster, produce the result faster. There is also the cost aspect. Illumina is cheaper than SOPHiA GENETICS for one sample. We also compared the variants between the two technologies. The detection of variants, both with SOPHiA and Illumina sequencing together with their own analysis pipeline, so SOPHiA platform and RAGN, plus the two sequencing technology with our in-house SARS-CoV-2 pipeline. Basically, what I show here is that there is not many differences between the methods. What's important here is that if we take the Illumina sequencing protocol together with our in-house pipeline, we were able to reconcile most of the observed differences. So we stayed with the Illumina sequencing and our internal in-house pipeline for SARS-CoV-2 for the workflow. Here is to show you how samples are processed now. Basically, this is what happens is in the lab, we get samples on Tuesdays afternoon. Samples are prepared, and on Wednesdays, there is the RNA to cDNA step and the amplification of cDNA. On Thursdays, there is the library preparation, and there is the sequencing that starts in the evening and run overnight. On Friday morning, we are able to do the analysis of the data and usually deliver before noon. There is an important constraint in this workflow is that we have maximum 12 days between the sample collection in the hospital and the GZ submission of the consensus. This is a constraint given by OFSP/BAG. So for example, HUG collects samples on week 1 from Monday to Friday, then the batch is sent to us the week after, and we follow the procedure depicted here, and we deliver the data normally before noon on Friday. So we are within this 12-day limit. But that implies that our pipeline must be the analytical processing must be fast because we don't have much time. So this analytical pipeline was not designed only for quality, but also for speed. And here is just to show you our current pipeline, which is described with the blue arrows in the middle of the slide. As you can see, it's not very fancy. We are just doing standard stuff. We use publicly available programs. And actually, our pipeline is also available on GitHub on the link on the bottom right of the slide. So we are within this pipeline within our computer infrastructure run in less than 4 hours, which was the goal for us. We provide different kinds of data to different institutions. We provide fast queues to Basel and since recently to CHUV as they do the analysis themselves. We provide the variants of concern and consensus in FASTA format to HUG, and HUG submit them to GZ then. And we also provide the sequences for genes and proteins to viral zone who do the phylogeny and the new variant surveying. Important point is that HUG is doing the submission to GZ themselves. Not all the consensus we produce pass the GZ criteria, as we can have frame shift. So in this case, the samples go back to us and we do some biocuration. And here I just want to show you 3 examples. The first one on the left, there is a deletion, frame shift 1 deletion that is reported by GZ as a problem. Actually, we confirmed by visual inspection, and this one, for example, was this frame shift was observed in many samples. In the central panel, you have a deletion of 22, which is a bit bigger than usual. Again, this one was confirmed by visual inspection, and the consensus resubmitted to GZ. Finally, not all the samples containing frame shift are confirmed, then like the last one. We have a deletion of 2 positions here, but the coverage is not sufficient for us to be sure about this. So the consensus generated from these bits will not be submitted to GZ at the end. And just to finish, sorry, there were arrows showing the mutations. So to finish, I just want to thank and mention all the peoples that have participated in these last 5-6 months in different aspects and giving us a lot of feedback. So thank you for your attention, and I give back the stage to Ioannis.
Hello again. The good thing is I'm vaccinated, so it's easy. We can be in the same room, at least me with one less than a mask. So what I'm going to do in the last 10 minutes and try to wrap up this webinar is to provide some guidelines of what we are staging in the next phase, and also trying to show you what we are trying also to achieve. The first one, which is the one that is the most important to the clinical community, is being able to annotate variants from the West, from the WGS, and more from the RNA-seq. The second part is an activity that we've been doing with the ETH domain PHRT related technologies, of which the Genome Center and the Proteomics and Metabolic Plex in Zurich are part of, and to try to explain how this is actually impacting and potentially changing the way we do clinical research in the future. I'd like also to highlight some very important aspects that are related to compressing the signal of genomic data, as well as privacy preserving. We have the luxury of having a very innovative landscape in the western part of Switzerland through the EPFL that actually enabled new technologies that we could really test bed. And I'd like to highlight that towards the end of the presentation. And finally, it's quite important that analysis could be performed on premises, so tooling and technologies that enable such kind of things for the researcher when they come either at our place or virtually is essential for the future of genomic medicine. Now, the variant annotation and clinical interpretation is really a multi-pronged approach. And we have the luxury of having enterprise and companies that have flourished in this area, such as Sophia Genetics or Safeto, that actually support different types of segments of the clinical support. But I think the Genome Center in that sense, and we should never consider a private and public partnership as something that is negative. It's actually rather an accelerator, but also a test bed. And the Health 2030 is definitely in a place to provide, depending on who asks, the different technology from these private partners. We have also companies like Congenica in the UK that are also active in that sense. And I think it's very important to us to be receiving the feedback from the people and the type of technologies they want to have implemented at the Genome Center, because ultimately we are doing this for the community. There are technology talking about community that have been developed some years back for the SHUV, for genetic disorder, called VARAP, that has been deployed on premise. And this is actually typically very useful and published in open source of technologies for variant classification in human genetics and Mendelian disorder. As Ilya showed you, we think and we hope that the DRAGene will also fit a bill in terms of time to delivery in annotations. I wanted to leave you with this idea that there is a landscape that exists. At the end of the day, people could decide which one we choose and which one is the best. But in the position of the Health 2030 Genome Center, we are there to provide an access that is to any of the one that is presented here, and that should be dependent on the vision requirement of our community. The second aspect, which I'd like to highlight, because I think it's important to also show what is the next step. You have perhaps realized that we haven't talked at all about amplicon, which is actually things that are implicated and run in hospital already. We are thinking 5 or 10 years ahead with whole genome sequencing, and perhaps not even West, although there is a good use of that. This project from the Kinderspital, from the Professor Matthias Baumgartner, in Zurich was really trying to push the boundary in terms of multi-omics and trying also to generate the data in a more systematic manner and try to digitize the sample into proteomic, metabolomic, genomic. The role of the Genome Center in that project was within a very short amount of time to turn sample into a digital form of a whole genome sequencing and RNA-seq. This has been achieved in less than 2 months, and it seems that there is a pattern. If I do the first Fourier transform, between January and February, there is something happened between the SARS-CoV-2 and this project a few years back. Let's see if it's every 3 years or every year. Now, in a few weeks, we had to also turn a first analytics, and that was thanks to both people in Zurich in the proteomics platform led by Bernd Wolscheid and Patrick Pedrioli, as well as the Genome Center, to both enhance the understanding of this disease, which is a disease of a rare disease, where we can actually explain more variation and reason for this disease to occur, but also quantify proteins and measure expression. As you know, clinical research is on a timeline that is longer than 2 or 4 weeks. So there is an intensive effort that needs to go on. This is actually what is the next phase of PHRT, is to enable a more systematic and more robust way of doing this multi-omics analysis. The outcome of this, and this is led by Patrick Forney and Caroline Frey at the Kispi Hospital, was to really take all this data and all the clinical data into a form that could be interpreted. And I'm just showing some ways to address and assess the data. And I don't want to go into the detail because we could look at the pathways. We could look at the metabolic scaling or the rate limiting step into the metabolism, because for each and every of these samples, we have a digital form of the sample in RNA, proteomic, and genomic. So that's what I wanted just to highlight with this example in a very few minutes. The second aspect, which I think is very important, as we know, genomics is generating more data than the Moore's law for computing, but also the law for storage. So enabling compression without losing data, so lossless, is becoming an essential stuff. We are very lucky to have EPFL pioneering a spin-out company called Genomics that just last week received an ISO accreditation for clinical IVD usage of what is called MPEG, which is the same as for movie entertainment, but in this case for genomics. And we got lucky to test this on our premises on an Alzheimer cohort of about 800 whole genome sequencing that is coming from the United States of America called Alzheimer's Disease Network Imaging. There is also another technology that we are investigating, the one that is implemented in a dragid called Emmanouil that Illumina bought some time ago. At this stage, I would like to thank the Genomics for an early access to the MPEG technology that we could test on our premises. And this is also one of the roles that we think the Genome Center could nurture, is also for companies and providing a test bed for companies that enable new technologies and innovation, which is one of the hallmarks from this western part of Switzerland that we live in. Another aspect, which is also a startup and a spin out of a very important aspect, we are at one stage depositing genomic data, which is the most sensitive data that we could think of, because it doesn't touch only ourselves, it touches everybody. So technology that are privacy preserving at the start and not as a thought afterwards is essential. Jean-Pierre Hubot at the EPFL and Jean-Louis Razero, who is now actually at the CHUV, have been deploying technologies from the medical community and deploying this so that basically the data do not leave the premises. In the privacy preserving, and our aim is to implement and test this for genomics, putting the bar at the whole genome and not the amplicome only, which is a much simpler problem. Interrogating the entire genome is the next future frontier in genomic medicine. Finally, and this is actually quite important, in order to engage the scientific community, we need also to have tools that allows enabling reproducible analytics in science. We are lucky to have within the ETH domain the SDSC that has been created, trying to create the middleware, as Olivier Verscher mentioned that a few years back. Trying to deploy this in our infrastructure is also essential, because we can in our environment, develop with researcher groups and implemented analytics that help, for example, the assessment of prediction quality. The genome laboratory is something that Ilya showed to you, but there are other projects that could be. And this is also in a very traceable manner, also transparency on the analysis that could be provided here. One example of an implementation that has been done, and I think we have never to forget that the PhD and master's students are the future of innovation, and they are the ones that drive the next companies and the next development, and also the next innovation, is the work that we've been starting with Jan Thomas at the HES in Yverdon, and Riet Wertenbroek, who is a PhD student now at the University of Lausanne, between Olivier, Jan, and myself, who worked really on trying to implement FPGA solution for CNVs, copy number variants and structural variants. We are actually quite adamant to push the FPGAs into the analytics. And I jokingly put to contribute to the Green Deal. And I think we have not to forget that all this is power consuming technology that we are doing. And anything that could allow us to use less electricity is something that we should really bring in our analytical framework so that basically we can continue to live on this planet in the right way. Sorry for being a bit philosophical here. That's my green touch here. Now, as a conclusion, I'd like to reiterate that the Health 2030 Genome Center aims to set, we have really a set of talents and expertise. I hope that showed you both in the accreditation that we just finished for sequencing clinical sample to the type of activity that the DIAP will run now. It's not a sequencing center only. It's a sequencing that enables analytics for both clinical research and diagnostic group in Switzerland. So if you want to come to work virtually or physically with us, there is both the emails and the Twitter, and we hope that this kind of presentation, even if they are remote and we can't have the discussion, which I hope we'll have soon in June at our annual meeting, will be actually continued and nurtured in the future. And with that, I thank you for your attention, and we'll look at the questions that are there. Thank you very much. All right. So let's see if we have a few questions actually. Some of them have been already answered while I talked, so that's good. So Michael is asking about CRAM versus the other compression. I think it's a very good question. The CRAM has been tested, and it's actually one of the portfolio. A lot of people are going in CRAM, but this assumes that you can't actually hide certain regions of the genome in a privacy-preserving manner. And I think we have to really take care of this. This is what Watson actually didn't want to look at its A-beta locus for a good reason. So I think we have to really test that. I think in our environment, the idea is really to evaluate all the technology that are there, both industry and academic, and try to move this forward for the community. But CRAM was tested in any case. I didn't mention it here in the slides.
Yes, okay.
Yeah, Michael?
No, the point was, I think that one of the big advantages of using EPIC-G or Genomics is if you dive deeper into their environment and what they provide, the whole stack, whereas CRAM is just the file solution. The question is, what is the benefit and the trade-off if you do the one or the other, because this can both lead to login and other problems.
So I think so there is one advantage, because the MPLAG I've been following for quite some time, the format to decode the data is open. And it's the same as for the video industry. The encoder is the things that people are putting there, blood and sweat, and it's signal processing in that sense. The decoder is actually something that needs to be absolutely open and clear. So there's no locked in that side. We have also to think, and this is where the Genome Center is located compared to the rest of the field, is really trying to cater for the clinical use. And the day you're getting into the clinical use, you get in a much more regulated industry-grade type of stuff, which, I mean, in this case, you might need to be in a locked-in, although you always want to have a way out. As we know, this is always a question. But this is actually the type of discussion that we have right now, is exactly the type of things that we would want to have more globally. And this is something also that we need to have globally with the SPHN and the PHRT landscape, because it's bigger than the small Switzerland, West Switzerland implementation that we are talking about. It's really to enable genomic medicine in that sense. So other questions that we have. So services in Switzerland are generally more expensive than other global options. Do you expect to offer sequencing to those outside of Switzerland, for example, to provide a greater level of privacy than might be expected from others, like Medco? So I think here we are open exactly the landscape in which Switzerland is for other fields that is not the genomics. People are trusting Switzerland for their institution, for their peace of mind and secrecy. So I think we might actually think of a role that is much more prominent in that landscape for Switzerland. At this stage, in the Genome Center, we'd like first to support the institutions that are internal to Switzerland, and then either through European projects or activities that would require the expertise of the Genome Center, move forward and step that up to a European or even an international settings. All right. Any other question that I see? Do you use deep variant or haplotype caller? Okay. Is there anything else that was not answered during the stuff? I'm just looking at it. Sorry for this.
Ioannis, I think an important question is whether we offer services for data not generated at the Genome Center.
Yeah, that's a good question. So the answer is yes, we can. We need to. So there's two things that we have to also see. We have to see that there are a lot. The computational biology and bioinformatics in Switzerland is a very advanced field. We have a lot of groups that develop methods. I showed you the group of Olivier Delano at the Department of Computational Biology. Michael Baudis at the University of Zurich is also one of the leaders in the oncology space for CNVs and others. I think it's quite important that we have to recognize that there are a lot of places where this could be done. Now, I would say the role for the Genome Center, the IAP, is that we could support this if there is not another group that could fulfill the mandate. And if there is actually a will to collaborate with us between an institution and us, that's even better, because I think we are always better working with different brains, and we should never be the most intelligent in the room in order to solve and crack research and clinical answer. Analyst?
May I say something on this too? I think that what is important to realize is that and Ioannis alluded to that, is that we're not trying to compete with any research group. What the Genome Center is trying to do is basically bring whatever has been created in Switzerland and elsewhere into a setting that can be done in a systematic way, business grade or whatever clinical grade, and high throughput. So if somebody has developed a software that they runs very well in their research setting, but they have no intention of offering services or setting up 20 collaborations with different people in order to run the software, the Genome Center can actually package it the same way and offer this as a service to be able to support the community that does not have the capability of running it themselves. Not for the point of making money, but actually for the point of providing that service through that. So, and that can be done clearly with a collaboration with the group that has developed the software. So essentially, we're more of a platform that allows for things to become much more, let's say, I wouldn't say business grade is one way to say it. Another way to say it is that to actually make them more streamlined, because research software is always something that you run in this clunky way to just get your analysis done, but then you can put it into a pipeline with other software to create something that is connected with other tools that might be used in a pipeline. So we are not intending to innovate in software development. The intention is to innovate in providing those solutions and facilitating the use of those solutions in an environment that also has the aspects of privacy, scale, and throughput, and all these things. So I don't know if I phrased it correctly for the sort of the computational terms, but You can sort of rephrase it if necessary.
I think that's a good point, which I didn't actually allude, is the fact that the implementation can go outside, and the implementation in terms of a running implementation could be done at Genome Center. This is a very important aspect. There is one last question I see from Jackie Beckman about, with the current amount of sequencing, how much could we do for the SARS-CoV-2? I should actually mention, because here we are only showing the Genome Center activity that contributes to the SARS-CoV-2. The DBSSC in Basel, as well as the Functional Genomics Center in Zurich, are contributing to this SARS-CoV-2 surveillance to around 2,000 samples per week. We have the capacity inside the Genome Center to sequence 1,500 samples per week, and the only rate limiting step is the thermocycler that we need to add for the PCR step. So in that sense, we are actually at a good number of samples that we could profile with this. Thomas has been asked a question and has been answered. And let's see, people tell me that there is 2 more. So it's the first time that we are doing a Q&A with almost 100 people. Yes, Olivier?
There are questions for Katrin, if you want to.
Okay. Which one it is? Regarding, so that's from Thomas Männik. Regarding the handling of metadata and sample annotation, we use SLIMs during the data generation process. That's what Katrin wrote. So
yeah, I started answering the questions that were directed to me.
Okay.
So I can briefly say that, yes, we are using SLIM system during the data generation process, so that basically each sequence that comes out of the machine also has an annotation and metadata report linked to it. And regarding standardization, that was the other half of the question. Yes, we do have a coordinated efforts with the BHRT proteomics and metabolomics platforms in Zurich for the multi-omic analysis, and we are also doing our contribution from Genomics side to the SPH and interoperability working group. So luckily we are working in a very well standardized field in genomics. So it's for the different efforts across the data sets, sure. And there is another from Michael Bowdis also to me, that was, where was the question? Will there be structured efforts to encourage staging of analysis projects for later researchers? For example, providing guidelines for forward-looking consent structures. Sure, we do encourage data repurposing and the secondary research use when it's consented so. However, as I said, we are not the data owners, and we do not also have the direct contact with research participants and patients. So this driving role should be taken by other institutions to establish a forward-looking consenting procedure. But wherever our expertise could be helpful, we are definitely ready to help.
Yeah, I think, in my opinion, it would be just good to say, to point, any project coming in to say, to kind of raise awareness and maybe point towards solutions, even if it's clear that the data ownership is clearly somewhere else, that the driver of the research are also somewhere else. That's completely understood. But sometimes really small things can be large obstacles later, and especially with consenting and making clear that the consenting is nicely done. That's good. And metadata, consenting and metadata, that's basically it, right?
Exactly, completely agree with you.
Yeah.
Thank you.
I mean, Michael, you're using one word which I think the Genome Center could play a role in, and I think other institutions are also trying to do this, is really to try to raise the awareness, because a lot of things are not known. Not everybody has been in a genomic revolution like Analyst and others, which is kind of the norm to do this. I mean, data was released as soon as it was sequenced. But other places where genomics become an essential part of the equation do not have that in their own DNA. And we need somehow to explain that better, and to also raise the awareness on the type of things that can and cannot be done. This is one of the roles, I think, of the Genome Center, but also other institutions and other projects in Switzerland that could pave the way for this. All right. I think we have done all the questions, if I'm not mistaken. I'm
So
very happy.
I see one question directed to me from
Okay,
Lucia. Yeah,
okay,
so there's a question about the turnaround time for in this particular case it's Wes, but it could be for any of the assays that we support. So we're in the process of developing a pricing policy for what we call fast turnaround time sequencing. That'll be published shortly. I'll caution that when you go from processing relatively larger numbers of samples with a given turnaround time to processing fewer samples with a faster turnaround time, economies of scale kind of work against you. So there could be an increase in the price per sample associated with that fast turnaround time sequencing. But we'll see when we publish that that list of prices.
Thanks, Keith. I think this is also where our next meeting, which will be in June, and just follow the Twitter, and we'll send that by email as well, will be also important, because by that time we'll be able to present and showcase the news. And by that time, I hope we'll get the beautiful page of the SAS for the accreditation that will come by fax, I'm sure. Analyst, do you want to leave us with a few last words? And
yeah, I just want to thank the whole Genome Center team for putting that presentation together. I think this is something that we intend to do more from now on, and keeping the whole community up to date with new developments, new services, and new manners in which we can interact with both clinical research community and other people that are interested in the services and support of the Genome Center. Our emails and phones and our brains are always open to any discussion that you might want to have on any project. And I think that I want to thank you all for attending, of course you, but I also want to thank the team, because they have all put together a great presentation, and they have done a great work up to now, and of course they will continue to do so, and I'm sure even more on basically delivering the mission of the Genome Center. I think that for those of you that have attended previous events and know a bit of history, the Genome Center started really small, started as a sort of an effort of some people and some ideas. And I think I can say that I'm particularly proud with what has happened, and the strong and amazing work of everybody. So there's more to happen, and it can only happen with the participation of the community. And we want that community, both clinical and research, to be driving the developments of the Genome Center, so the Genome Center remains relevant to all of you, and of course, to be able to support the development of genomic medicine in Switzerland. So thank you all for attending, and we are very close to you, whatever you need, whatever questions or concerns you might, or needs you might have.
thank you, Manolis. Thanks to everybody. So the slides and the recording will be distributed so that people can consume it at their will if they want to spend more time behind the computer. And I wish you all a good lunch, and see you soon, hopefully in vivo and in toto the next time at the Genome Center in Geneva. Wishing you a good day. Bye-bye for now.